Thanks very much. Okay. You guys get through. So next up, again, to speak to the sort of the broader holistic One Health aspect, we've got Tim Johnson, who's an associate professor at the College of Veterinary Medicine, University of Minnesota. Tim works on spread of antibiotic resistance through gram-negative organisms, and is going to speak to us from the animal side.
Well, thank you. I appreciate the opportunity to be here. And the strategy I'm going to take is a little different. I'm going to give you some success stories in animal agriculture about alternatives that I think are working. But I'll start with the disclaimers, which number one is that in terms of what we know so far, there's no single replacement for antibiotics and what they can do. So what many people have been looking at, and what I'll highlight, are some strategies for combinations of products. And I'll mention some specific ones, but I'm not endorsing any products. These are based on extensive discussions with veterinarians in the poultry industry about what's working and what's not, and then scientific literature that has some science behind it. So how many of you have gone into Walmart or a grocery store to look for probiotics and encountered this and said, okay, which one do I pick? And then said, okay, well, the most expensive one must be the best one, right? Well, I've had that problem. Or if you go to the yogurt section and say, okay, well, which yogurt has the best probiotic in it? Some have claims about what, regularity and all these other things. So it's tough. And if you imagine poultry, obviously we're definitely interested in the microflora, and gut health and the microbiota are paramount to keeping flocks healthy. And you can imagine that if you were faced with decisions of dozens of different potential alternative products and sales reps coming to your door telling you mine is the best, or no, mine is the best every other day, it's challenging when there's not a lot of science to back these things up. So that's what veterinarians are faced with on the animal ag side, is trying to determine what products do I use when there's not a platform or there's not a central location that you can go to and find out which ones work under which situations. And this isn't a new idea about alternative products. If we go back to 1973, a person named Nurmi, a scientist, realized that if you take an adult microflora or an adult gut content from a hen and feed it to newly hatched chickens, what you find is that it will inhibit or reduce salmonella colonization. So this concept of using the microflora, the self-microflora to combat pathogens isn't a new idea at all. But if you look at what's out there in terms of products, there's a huge list, and I don't have time to go through all of these. We could have a couple of hours to do this. But typically speaking, we're looking for things like prebiotics, probiotics, so feeding the gut, seeding the gut. There's also alternative products, so oregano, saponins, garlic, thyme. I mean, it sounds like somebody made the comment that we're actually marinating the chicken by giving them all these things. And I say, does that actually change the flavor of the chicken? No, it doesn't. But a lot of alternative products are being explored right now, and this is an interesting time in animal agriculture, because with the veterinary feed directive and this transition to many alternative products, it's really a tough time to try to figure this out. Oops. And so I'll just give you a few of what I see are interesting stories, so things that are a bit different in terms of what people are finding with use of alternative products. So one of these, so this is a prebiotic called XPC, and so it's a combination of extracted nutrients and things to basically feed the gut, and this is a widely used product in the poultry industry as well as other animal ag industries. What's interesting about this product is, and kind of unexpected, is so the product was geared towards improving feed efficiency, helping with weight and performance in birds, and also reducing pathogens like salmonella. So there's some good data, an extensive list of publications showing that it will help reduce the colonization of salmonella in the gut of the animal. But what they found with additional studies, and quite extensive studies actually, is that these products are also appear to be reducing resistance of salmonella. So they did a survey of several thousand samples over hundreds of different flocks that have been treated with this product or a control, side-by-side control without the product, and found that the salmonella that they were isolating, while they were reduced in number, were also less or more susceptible to drugs, less resistant to antibiotics. And it turns out that there's been some mechanistic studies done on this, and what they found is that it's actually expelling or causing the expulsion of one of the genomic islands of salmonella that contains multi-drug resistance in it. So I think there's a lot of kind of coincidental findings here that tell us that there's more to this story, and that there are products out there that could have the potential to have an impact on resistance. We just don't really know the mechanisms yet by which some of these are doing that. You've got probiotics and prebiotics, and I'll talk about probiotics in a bit, but we also have something that we don't think about as much, which is vaccines. And so there's definitely commercial vaccines out there, and most of them are killed vaccines where we find the right bacteria and either kill it or even attenuate it and sell it as a commercial product. But we also have subunit vaccines that are being used in animal egg, and this is an example of one that's been very effective. It's got some pretty broad cross-protective qualities, and it's based on the iron systems of the bacteria. So iron receptor proteins or siderophore receptor proteins. And this technology has been used in a variety of different vaccines and shown that it really can have an impact on the occurrence of disease in the animal. And you can see it's cross animal species too. So we've got chickens with cholera, fowl cholera. We've got mastitis in cows. And in even humans, it's being applied as a model to prevent urinary tract infection. But you know what I think is even more interesting is the use of autogenous vaccines. And so an autogenous vaccine, if you don't know this, is if you don't have an effective commercial vaccine that you can use and buy to use against the disease in an animal, you can make your own. So the veterinarian can identify strains and send them to a company that will produce the vaccine directly from the strain from your flock, and then you apply it back to your flock. And this has been highly effective, but the main problem with this has been it's difficult to survey pathogen ecology. So we didn't have the tools previously to figure out what strains would represent the most effective vaccine. And so with genomics now available, cheap, fast, many companies are starting to do more detailed surveillance of isolates. I'm not going to try to explain this heat map, but it's based on SNPs and our single nucleotide changes in genomes. And the story of it is, just to give you an example, that here's a company that had a producer that couldn't solve a Salmonella typhimurium problem, even though they were using a Salmonella typhimurium vaccine. They went in and looked at the strain composition across their system and found that the strains that were there weren't similar to the vaccine strain that was being used. Then they took one of their own strains, created the vaccine, and solved the problem. So I think that the ability to use these techniques, and this is being used, beginning to being used widely in animal egg, this idea of pathogen surveillance. And we've done a little bit of this with another bacteria called, one that I promise you 90% have never heard of, Ornithobacterium rhinotracheale, or ORT. So this is a respiratory bacteria that causes disease in turkeys. And I put this up here because this is an interesting story. So each color is a different company that was using a vaccine, and we looked at their field isolates. And so the boxed colors are the actual vaccine strains that were being used, and then the other strains that are in the same color are the field strains. And you can see in all cases, the field strains don't line up in the phylogenetic tree with the vaccine strains. So that's good, because the vaccine is pushing the pathogen population away from whatever it is that they were targeting. The bad thing about this is they were continuing to get ORT infection with other strains from different lineages. And an aside to this is that up at the top here, this what looks like what we call monophyletic clade, or a clade of very closely related isolates. That's basically the same strain that's been around for 30 years that I found out through discussions they've been actually seeding their flocks with to try to induce immunity. So these types of studies can tell us a lot about what strains, what multiple combinations of strains should we use to vaccinate, but also what's you can use it as a diagnostic tool to figure out what's going on in a flock. So other custom approaches to developing probiotics. I think that this is where animal ag is going. It's not simply buying a probiotic and just continuing to use it. Now what's going on is that we're actually seeing people sampling isolates or pathogens from their flock or herd and looking at those pathogens in the context of, well, if we have a library of potential probiotic strains and we've got a library of pathogens from our flock, can we identify the right combination of probiotic pathogen to start getting rid of some of those pathogens? And this is an example of one company that's been doing that, where they'll take avian E. coli, which is a big problem in poultry E. coli infections, and they'll characterize them with genetic techniques, and then actually take a Bacillus probiotic library and look for strains that are going to be effective against the pathogen. And so this has been a highly effective way to reduce E. coli load in the flocks of poultry that have problems with E. coli infection. But I do want to say there's a cautionary story here, a message, and that is that what we've seen in the field is that, so this is one example, a company or a producer that had been using it's a combination of 5 strains of Bacillus, been using it for about 3 years, and green is susceptible, red is resistant, and that's the pathogen being resistant to the probiotic. And what we see happening is that over time, even with strain cycling, you're still seeing resistance developing to the probiotic strains themselves. So I think that there's a message here that we need to be careful about these alternative products that have effect and not kind of doing the same thing that we've done with antibiotics, which is allowing resistance to develop over time. So I'll finish with the microbiome. And so I mentioned gut health, and certainly you heard in the last talk that gut health and the microbiome is an extremely important part of trying to understand if a product is having an effect, if an alternative is going to be useful. And so this is a PCOA plot. It's a plot that shows you all kinds of different gut samples. So we've looked at chickens and turkeys and looked at about 10 to 15,000 different gut samples from chickens and turkeys and tried to define what is the base microbiome and then go from there. And so this is just looking at the ileum of a chicken in different samples. And the point I'm trying to make here is each dot is a microbial community from an animal, right? And so the closer they are together, the more similar they are. The farther apart they are, the less similar they are. And what you see is that we see very predictable patterns. I should mention that this is looking at 6 different flocks of chickens from 6 different companies. But we see the same pattern of succession. And succession I mean by day of age, which is over here. So there's a predictable succession of the microbiome over time. If you look at by tissue, like cecum, ileum, or even trachea, different sites in the bird, predictable pattern there. So there's a lot of predictabilities about the microbiome of an animal. And that's a good thing because we can know what dominant bacteria are there and we know what we need to modulate. The problem is when you start looking at treatments, we haven't seen a lot of impact. So we've looked at a variety of different types of products like the ones I mentioned earlier. So prebiotics, probiotics, essential oils, all these different things. And this is typically what we see when we're looking at control versus treatment. And you're looking for clustering of colors, right? And you don't see any clustering of colors here. I'd say that 95% of the products we're looking at aren't really having a major impact on the gut microbiome. And it's not just all about the gut microbiome, but that's certainly an important piece to it. So we also look at the gut villi and how the gut develops, and then of course performance. But this got us thinking about, well, what can we do to modulate the microbiome? So what we've been doing is to try to look for, take the probiotic approach a step further and start thinking about host-specific probiotics. So probiotics that are sold to poultry right now, there's, like I said, dozens of potential probiotics that you can sell. I believe there's one that is actually a chicken-specific probiotic, and the rest of them are probiotics from other sources. There's no turkey-specific probiotics combinations. And so we started looking at, well, if we take strains out of the turkey gut and characterize them, is that a better combination of strains for a probiotic than if we just use chicken source or other source? And what we're seeing is that we can actually see changes. So we compared it to an antibiotic. These are controls. This is the chicken source probiotic, and this is the turkey source. And we see that both the chicken source and the turkey source, when you give it to the animal orally, will have an impact on the microbiome. If you look at performance, the turkey-specific probiotic is the only one that has an impact on performance. If you look at bacterial diversity, the turkey-specific probiotic increases diversity in the gut. And then I think the big piece of this is starting to look at the host as well. So we've started to look at the host and see how many genes are changed in the host tissue when you give it this probiotic. And that's where you start to really see the patterns. And that is that an antibiotic changes a number of genes in the ileum, whereas the turkey-specific probiotic changes a moderate amount, whereas our chicken-specific didn't really change any. So I think there's this microbiome host connection that's important to study. And so I'll just finish with knowledge gaps. And I think that, we know a lot about what works, what doesn't, but we don't know the how behind it. So I think there needs to be more mechanistic studies about how these things are working and why they're not working, and how does antibiotics compare to other products. There's a lot of combinations and possible products that haven't been even explored yet. Another point is that we can't translate what's happening in the turkey to the chicken or the pig, or even when you're looking at genetic lines, we see differences between animals. So all these things have to be taken into consideration. So it gets very complicated very quickly. And this timing and under stress, we don't really know much about how these products work when you put the animal under stress, and then which ones are working when. So I'll finish there and just want to leave you with a picture of Minnesota and where a lot of these industry collaborations take place. So thank you.